Greetings to everybody who is watching this. We'll discuss the first chapter of our biotechnology, and this is the book, if you don't know, in case. This is the biotechnology class 12th and textbook for class 12th. And mind you, this particular chapter, the first chapter of this book, an overview of rDNA technology, or also known as genetic engineering, is a chapter which is usually not asked in the school examinations or the periodic assessments you will go through. This is a chapter just for overview, a bird eye view. We are just skimming above the surface, and actually, we are gonna delve deeper into the nitty gritty, the kernel of the topic, the subject in the upcoming videos, most probably. This is relatively a new subject in the study arena, so to speak. So, this is the biotechnology textbook for class 12th, and there are many chapters in this. The last two chapters and even four chapters, in my case at least, they were not asked in the examination, which are entrepreneurship, and chapter 11 and chapter 10 is bioprocessing and biomanufacturing, and the 11th one is bioremediation. These chapters were in my section, at least in my case, in the boards, were not asked, and we are discussing the subject as there are almost no videos, study materials available for you on the subject on the internet. So I just thought, let's do this. And whatever I have understood, whatever I have assessed and found out in the subject, I'm just trying to share it with you all. So without any further ado, let's start the chapter. So an overview of rDNA technology. We all know that we slowly did our evolution. We slowly adapted. We slowly found out new things in the field of science, in the field of biotechnology. It gradually developed over time. It augmented over time, one after another, brick by brick. So we all know that Frederick Michaud, who was a scientist who discovered nuclein inside the nucleus of a pus cell from a discarded band-aid, and then another scientist came known as Altman, who described the nature of these nuclein components formed, and he said that these are acidic in nature, and that's how the term nucleic acid came into existence. So in the last century, scientists discovered that nucleic acid, here in this case DNA, is responsible for the expression of characters. Now, this was a huge hallmark discovery. This was the cornerstone in the biotechnology, as this discovery led us to the genetic makeup of organisms, the genome, and then how can we manipulate that? And there were various techniques and procedures which were developed, and they were all kept in a single bracket, which was known as genetic engineering or the rDNA technology. The first breakthrough in this journey, as it is mentioned, the DNA is not only the element responsible for carrying the genetic information, but it is also the element responsible for propagating it. And this is a cherry point. This is the strawberry point for us. This is a point to remember. This is a really important point. I mentioned that this rDNA technology or the genetic engineering is the name of the same thing. Naam anek, banda ek. The first breakthrough in this journey was establishing that DNA not only carries genetic information, but also, this is the cherry point. I can't stress on remembering such statements in the exam. It is very handy. You can use them. And this really conveys a huge thing in just a simple statement. So it's an important statement. During this period, scientists, researchers observed that bacteria is able to take the exogenous DNA, the genome from surrounding. And when they observed this particular phenomenon, they said, why can't we introduce some genes and why can't that bacteria integrate that into its system? And that's how the whole discovery, whole demonstration came into existence. And that's how they asked the question that is it possible to transfer the interest gene, the gene of interest, that insert in the organism to obtain its product? And now there are two scientists which come in the picture. So the first scientist, Stanley Cohen, he had expertise in introducing plasmid DNA into Escherichia coli. Escherichia coli is a bacteria which multiplies very fast. So it multiplies, if I remember correctly, in 20 minutes. So in 20 minutes, it is actually able to reproduce. And Stanley Cohen had this expertise, had this proficiency. He was an adept in introducing plasmid DNA into the host. And Herbert Boyer, who was another scientist, and he was his partner later. So they formed an alliance, they formed a union, they formed a partnership, a camaraderie. So he was the another comrade, and he had the expertise. He was proficient. He was dexterous in restriction enzymes. With restriction enzymes, which are just the enzymes to cleave double-stranded DNA. It cleaves, it hydrolyzes the phosphodiester bond. In case you are wondering what is a plasmid, what are these vectors, and what is this all about? So shortly, I would mention that plasmids are a kind of vector. What is a vector? Vector is just a vehicle, to put it simply, and this is just used to transfer, just like a delivery boy transfers goods from one point to another. Similarly, the plasmid is a kind of vector which does the same job. They both formed an alliance, and then their discoveries, they combined together and they led to the rDNA technology or the genetic engineering. Now comes what is a plasmid. Plasmid is a double-stranded, circular, and extra chromosomal part of bacterial genome which is able to self-replicate, and this ability makes it useful in genetic engineering. So plasmid is a distinct structure, a discrete structure, and that is able to reproduce itself autonomously. So it is not dependent on the bacterial DNA, the main DNA of bacteria. So that's why it is used in genetic engineering, as we need a vector which is able to multiply itself after going inside the host organism. Now, organism associated with plasmids are that it is found in the Monera kingdom, in the Mycoplasma, in the PPLO polypneumonia-like organisms, and also in the bacteria, which are the most common. Plasmid in genetic engineering. Now, what is the role of this plasmid? So it is a vector, and there are some distinct characteristics of vector which we will discuss in the later chapter, in the second chapter. For now, let's say it's just a vector and it is having the nature of self-replication. That's why it is used in rDNA technology. rDNA tech contributions. Now I remember that in my Viva Watch, in the biotechnology interrogation section, the examiner asked me, tell me some contributions of rDNA technology in our daily life. So these kinds of questions, they appear in front of you in your Viva. So make sure that you do this. And this is the first and the foremost thing you must know before studying biotechnology. What is the applicational usages of it? rDNA technology contributions. It has played significant role in shaping our life standards. For example, treatment of various diseases, various genetic disorders are treated by the help of rDNA technology. It has contributed in improving, improvising, and developing disease-free, high-yielding crops, which are so. Have you ever heard about the term HYV seeds? I remember in high school or in class ninth, we studied in social sciences HYV seeds, which are high-yielding variety seeds. And this is an example of purely a biotechnological application. Now, examples of rDNA technology in medicine, you see that there in the erstwhile, in the previous century, people used to slaughter millions and millions of animals. Tons of animal pancreatic glands were needed for producing few milligrams of insulin to treat diabetic patients. And if I remember, there were two kinds of animals used. One was the porcine insulin, and another was the bovine insulin. Bovine is for cattle, and bovine and porcine. Porcine is for the pigs. So these were the two animals which were used, and but there were several disadvantages in this. Earlier, tons of pancreatic glands from animals were needed to synthesize only a few milligrams of insulin, which was obviously economically not feasible for the common folks. And that's why we needed new technologies by which we can develop the insulin, growth hormones, and other therapeutic drugs easily, straightforwardly, and in a very simple fashion, not in this complex fashion, and that too with the slaughtering of millions of animals. And thousands of pituitary were needed for the synthesis of growth hormone. These products were in limited quantity and were very expensive. Additionally, purified therapeutic proteins from animal sources were often used. And they also induced some immunogenic reactions in the diabetic patients, as there were some differences in the amino acid placements which contributed into the immunogenic reactions in the humans, because at the end of the day, what is an insulin? Insulin is a polypeptide chain, and there are amino acid sequences. Now, if you don't match those completely with the human insulin patterns, it is obviously going to induce some immunogenic reactions. Now, scientists had some challenges. but they overcome it, and that is human insulin and the growth hormone for treating dwarfism. So these were synthesized through rDNA technology and creating interferons to treat cancer. We'll come to what is interferons in the next slide. Plasminogen activator and urokinase are used to dissolve the blood clots. So rDNA technology in agriculture, it is used to synthesize crops which are better yielding, which are high in productivity and quality, and also they are resistant to several pesticides, insecticides, rodenticides, resistant to several groups of insects, and high levels of abiotic and biotic stresses. So these are advances in the agriculture. In the case of rDNA technology, we can introduce the target modifications and we can genetically produce modified crops. Now, this has resulted into crop resistant to diseases, helping farmers to avoid crop damage, development of drought resistance, the abiotic stresses, and salinity tolerant, so pH tolerant, allowing farmers to grow them in harsh environments. The impact of rDNA technology is that it increased the quality and productivity. Tell me one thing. As the human civilization is exploding day by day, as we are seeing, we need better yielding crops to deal and to suffice the hunger of such an enormous population. So that's why we need these technologies. Now, interferons. In the previous slide, this was the term which was written. These are the proteins produced by the body's immune system, and they are also used to treat cancers because these are immunogenically produced proteins and are used in the cancer treatments. So we isolate these genes, the genes encoding for this protein, then we insert this in a plasmid, and after that we introduce in the host system for proliferation. Now, plasminogen activator is a protein that is a clot buster that dissolves blood clots, and we also again we isolate this, we insert this in the host, and then we multiply this, we collect this through the downstreaming process, and then we actually make this for human use after several trials and tribulations. So this is used to treat patients with heart attacks and brain strokes. Now there are strokes which are of two kinds. One are ischemic and another is hemorrhagic. So hemorrhagic is only 30%, rest 87% is ischemic. So this is caused by the blockage of the blood flow, which is the maximum case. And hemorrhagic is when there is something that ruptures. When blood vessels rupture, that is a hemorrhagic stroke. What are urokinase? Urokinase is another protein that helps in the blood clot. It helps in dissolving the blood clots, and that's the work of it. Which is the host, we collect the protein from it after our downstreaming process. That's the myocardial infarction, another name for heart attack, occurs due to the blockage of blood flow. So these are some dates which is up to you whether you memorize this or not. Because personally, I'm not in the side of mugging up these details, dates. It doesn't contribute in any learning. We had to do this. These are some important ones, which is Carl Ereski coined the term biotechnology in 1917. Avery, MacLeod, and McCarthy in 1944, they demonstrated that DNA is the genetic material. They took 3 test tubes in which RNases, DNases, and proteases they added, and then in the test tube where DNases was added, there was no propagation seen. So that's how they concluded. Watson and Crick proposed the double helical structure or prototype of bDNA, which is the most commonly occurring DNA. 1970, Werner Erber. So this is the main name, which is Matthew Messelson, who discovered type 1 restriction enzymes. And 1973, Stanley Cohen and Herbert Boyer developed the rDNA technology. 1983, this will come later onwards as well in the polymerase chain reaction, which is the molecular photocopying machine. 2003, Human Genome Project was completed, and recent advances are in CRISPR-Cas9 genome editing tool. If I remember correctly, the full form for CRISPR-Cas9 is the protein which goes with the rDNA and cuts the site of the viral genome. CRISPR is clustered regularly interspaced palindromic repeats, which is a technology we will discuss in the upcoming videos. Now there are question answers. The first question is, how was rDNA technology initially developed? This you can do by yourself. Then application of rDNA technology in crop improvement and therapeutics has contributed to development of crops that are resistant to pests, diseases, and environmental stresses, which are abiotic stresses. We have developed genetically modified crops that are produced by inserting desired gene into the plant genome. The therapeutic uses is developing vaccines, growth hormone for treating dwarfism. Achondroplasia is also a disorder which is in which we use the growth hormone therapy. Who discovered plasmid? Joshua Lederberg. Plasminogen activator and urokinase are used for blood clotting. This is a blood clotting dissolving drug. Plasminogen activator and urokinase, which are the two proteins we already discussed. This is the assertion, which is restriction endonuclease cuts the DNA and is isolated mostly from bacteria. That's absolutely correct. Reason is restriction in endonuclease is a type of nucleases, which is again a really correct, cognizant, logically sounding explanation for it. So that's why option A. Question 2 is assertion, which is E. coli divides in 20 minutes while replicating its DNA in about 60 minutes. The reason for this is E. coli. Escherichia coli follows a multi-fork replication mechanism where different regions replicate at the same time. The assertion reasons both are correct and logically sounding, and that's why the option A is correct. So my final remarks on this chapter will be about the enzymes in general. So enzymes in a very generic sense are of several types. For example, there are transferases, isomerases, proteases, amylases, chitinases. Chitin is a particular element which is found in the chitin, which is in the cell wall of the fungi. So to digest that, you need chitinases. So there are different kinds of enzymes. There are hydrolyses in which there is a category, a subcategory, which is the restriction endonucleases. Now this comes under the nucleases, which is another category, which is the general category under the hydrolyses. And the nucleases have several types of the enzymes in its subdivision. And the categorizations are like restriction endonuclease and restriction exonuclease. As the term suggests, that exonuclease, which is that it takes the nucleotides from the outer periphery, from the marginal sites, from the fortifications, from the boundaries, whereas the endonuclease, as the term suggests, as the diction suggests, that it is the type of restriction enzyme that cuts the DNA elements into two pieces from the middle or from whatever the sequence it founds as a palindromic repeat. So the sequences which are palindromes, it will cut those parts, those specific elements of the DNA. And that's how the restriction enzymes work. Now there are two kinds of restriction enzymes, as I have said, restriction endonuclease and restriction exonuclease. There is also one more thing here, which is a catchy point, and that is whenever the nuclease hydrolyzes the phosphodiester bond, there are two possibilities, two plausible occurrences which we can see. The first one is the formation of the blunt ends, and the another is the formation of the sticky ends, or also known as the cohesive ends. So cohesive ends is like in this fashion, whereas the blunt ends are like straight away. So this is just a simple outline on what the another chapter comprises, and that's all for now. Let's call it an end. Thanks a lot. Thanks a ton for watching this video if you have watched till this point, and let's call this end. Thanks for watching.